This presentation considers how general intelligence is measured. It also goes on to consider how the heritability of general intelligence can be studied, focusing on two methodologies: adoption studies and twin studies. These methods are important historically, although they are gradually being replaced by other forms of study. The statistic for general intelligence, G, is derived from the results of a series of independent tests for different cognitive abilities, such as reasoning, processing speed, memory, spatial awareness, and so on. The final value of G is derived by factor analysis, in which the results of individual tests are weighted relative to the importance of the domains chosen. It's important to recognise that there is a variety of different approaches to this, and that there is no one single method for calculating G. The Wechsler Adult Intelligence Scale, WAIS-4, is probably the most widely used intelligence test in the world for both adults and for older adolescents. It can be used to establish a measure of G. The diagram shows the structure of the various domains that are used to produce an IQ score. FSIQ stands for Full Scale Intelligent Quotient and is an estimate of G. Because the estimate of G is based on the weightings of other factors, it is regarded as a second-order factor. Recent developments in the methodologies underpinning the test are trying to develop ways of developing estimates of G directly from the various subtests. This is called the bifactor model. A person's estimate of FSIQ is compared with a standardized sample of 2,200 people in the United States, ranging in age from 16 to 19. The final estimate of G, the FSIQ, is made by factor analysis. When a population of G scores is compared, it shows a normal distribution. G accounts for about 40% of the differences between individuals taking the tests, which makes it a very important factor. It is important that we realise that it does not account for all of the differences in the distribution shown. Nonetheless, it is possible to rank individuals in the population and then to relate this score to the range of intelligence shown in the whole population. This is one of the most important contemporary human geneticists in the world. It is Robert Plomin, who works in London, although he was originally born in the United States. Plomin argues that little g, as estimated by tests such as WAIS-4, is one of the most reliable and valid measures in the behavioural domain. Its long-term stability after childhood is greater than the stability of any other behavioural characteristic, he argues. And Plomin is not alone in this view. The study of general cognitive intelligence is a fertile one for writing assignments on this course, and there is a significant amount of background literature for assignment writing. The slide shows two such sources which are in the library. We are now going to consider how to study the heritability of G. That is to say, the proportion of the differences between people in the population which can be attributed to inherited or genetic factors. To do that, we need to apply a simple three-factor model to the variation in the population. This is known as the ACE or the ACE model. A measures the proportion of the variance which is passed from parents to children. That is to say, genetic variation which can be inherited. The effects of these different genetic factors are all combined together to produce an overall effect, and therefore it is called additive genetic variation. Non-inherited effects are attributed to the effects of the environment, and these are divided into two subgroups: shared environmental effects within the family and non-shared environmental effects which are unique to a particular individual. The next thing we need is the degree of relatedness between the individuals within our population samples. If we work with pairs of identical twins, then we expect them to have 100% relatedness. That is, they share 100% of their genetic information. This is something we will consider further later. Non-identical twins, and indeed any other first-degree relatives such as brothers or sisters, would be expected to have only 50% of the genetic information in common. Thus there's a relatedness of 50%. If we compare a parent with their offspring, then we would also expect 50% of the genetic information to be the same. Comparing grandparents with grandchildren reduces the genetic similarity to 25.5%. Not surprisingly, comparing unrelated people gives a relatedness score of 0%. It is important that we realise that these studies are looking at the differences in genetic factors between people. The fact that 99% of the genome is the same in all humans does not feature in this analysis, which only concentrates on differences. This is an important slide which is worth studying carefully. One way to study the heritability of cognitive abilities is to use adoption studies. A child is adopted into a family and therefore experiences the same shared environmental effects as the other brothers or sisters in that family. However, those brothers and sisters also share a genetic similarity with their parents, which the adopted child does not have. By studying families in which some children are adopted, we can study three different effects. We can look at the similarity between the biological brothers and sisters which are sharing both genetic and family environments. This is the genetic plus environmental graphs on the chart. We can also compare the similarities of genetically related family members in an adopted non-shared family environments. Sometimes we can compare an adopted child with its biological parents or with any other biological brother or sister which have been adopted into different family environments. Finally, we can compare the similarities of non-genetically related family members who are living in the same shared family environment. If we look at the chart, we can see that adopted offspring show a greater similarity to their biological parents than their adopted parents. Look at the figures 0.24 in the middle genetic column for parents and offspring, and compare that with the figure 0.20 in the right-hand environmental column for adopted parents and offspring. The sibling correlation in the environmental group gives a measure of the shared environment. The genetic plus environmental column is effectively the control group. Siblings brought up with a shared family environment with their biological parents show a correlation of only 0.47. That is, only about half of the differences between them in their test results can be explained by genetic and shared environmental factors. It's important that we understand what these results show. We are not finding any evidence of genetic factors completely controlling the development of children in families. And equally, we are not finding any evidence that shared environmental factors are the only determinants of children's developments. It looks as if it is nature and nurture, rather than nature or nurture. And there's still plenty of room for individuality. However, we cannot say that unique environmental influences are the only factor that shapes development. What we're looking at here is an interaction of genetic, shared environmental and unique environmental effects. This is an idea that is at the heart of this course, which we will return to regularly throughout the rest of this course. Adoption studies are a very powerful way of trying to separate the effects of the inherited and non-inherited components in the differences between the test results for family members. If we're going to generalize these conclusions beyond the studies, then we will be making a series of assumptions. We can outline some of these assumptions here and then develop them further later. The first assumption is that parents and children involved in adoption studies are typical and representative of the rest of the population. The second assumption is that when a child is developing inside her mother's uterus, any effects of the environment on her growth will be correlated with the mother's genetic effects. This blurs the separation of genetic and environmental effects between the biological mother and her offspring. Likewise, any social contact between the adopted child and its biological parents also blurs the separation of the environmental effects between the adopted family and the biological family. If any of these effects are significant, then this will reduce the generalizability of any results from adoption studies. The second way in which heritability has traditionally been estimated is by using twin studies. The next two slides consider how identical and non-identical twins form. Identical twins are better called monozygotic, or MZ. This is because identical twins are not necessarily completely identical in all of their characteristics. Monozygotic twins are formed when a single egg is fertilized by a sperm. The fertilized egg divides normally many times to produce a ball of cells. These separate into two embryos and develop into individual twins. All MZ twins are assumed to have the same genome, although it is possible that some mutations might occur after separation, while they're still developing in the uterus. Therefore, all MZ twins are assumed to have 100% genetic resemblance. All babies develop in the uterus connected to a placenta, and in MZ twins about 30% of them develop from the same placenta. About 66% of MZ twins develop from separate placentas. It is worth thinking that twins inside the same uterus may or may not be getting comparable supplies of nutrients and space. Indeed, there is some evidence that twins compete with each other for these limited resources. Non-identical twins are called dizygotic. This is because two eggs are released from the ovaries and are then fertilized by two sperms. DZ twins share on average 50% of their genetic material. They always develop with separate placentas. DZ twins have the same relatedness as any first siblings and can be different sexes. Twin studies usually use the same sex DZ twins in order to be able to compare them with monozygotic twins, which are always the same biological sex. This is a complicated slide which compares the results of a variety of different techniques for studying the heritability of general cognitive ability. It's looking at the genetic correlation between parents and offspring and between siblings in the different types of design. The correlations between adopted children and their genetic parents is 0.24. This is also the same for any other first-degree brothers and sisters who have been raised apart from each other. The degree of genetic relatedness in both cases is 50%, and we double the correlation to estimate the heritability, which makes it 0.48. This suggests that about half of the differences in general cognitive ability in the study would be due to genetic factors. The remaining 52% of the differences are due to the effects of the environment. We cannot separate shared and non-shared environment in this particular study. When we look at the twin studies, there are two kinds of studies. A direct comparison of differences between pairs of MZ twins and pairs of DZ twins. We can also, in the adoption study, look at comparisons between MZ twins who were separated at birth and then reunited in later life. Such studies are quite rare, and in this analysis are divided into older studies and newer studies. A comparison of the twins who are raised together are on the far right of the chart and show that MZ twins correlate with each other at 0.86, whilst DZ twins correlate at 0.60. The difference between the MZ and DZ correlations is 0.26. Doubling this gives a heritability of 0.52. The correlation of MZ twins reared apart gives a direct estimate of heritability. Newer studies are giving a heritability of somewhere between 0.72 and 0.78. We can use model fitting techniques across all of the data to give us an average heritability of about 0.5. That is to say, about half of the differences between individuals in these studies are due to genetic factors, and about half of the differences between individuals in the studies are due to environmental factors. Similar results are obtained with studies across the world, and this is often used as a justification for the reliability of the techniques and the validity of the analysis. It is important to realise that all twin studies make a number of assumptions. Twin studies assume that the shared environment is roughly the same for MZ and DZ children raised in the same family. In other words, parents of MZ twins treat their children the same as any other children. It also assumes that twins share a common environment in the development in their mother's uteruses. Violating these assumptions inflates the estimate of the heritability of G. If we can generalise these findings, and Robert Plomin is confident that we can, then about half the differences between people for G are due to genetic factors, and half of the differences are due to the environment. As Plomin says, G predicts key educational and occupational levels better than any other trait. Genetic factors do play a part in G, but we are not pre-programmed robots. This tells us nothing about the interplay between genetic and environmental factors in our individual development or in our future biographies. Equally, we cannot be naive enough to pretend that genetic factors play no part.